How do we translate the information from messenger RNA through transfer RNA? So, for instance, if on the messenger RNA we have the triplet codon AUG, we will use the decoder. Notice the first base A, second base U, and then the third base G, AUG, or the start codon. Let's look at ACU. AUG, the amino acid, ACU. So here's our first bit, A, second C, ACU represents 309, the amino acid, THR, and there is your THR. What about AAA? We use the decoder. A, first base, second base A, and there's your AAA, which represents lysine, the amino acid. So this means, so what is the process? How do we eventually get the amino acids? We use the decoder, then a bond between the amino acid is the peptide bond, which eventually builds the protein. All right, so basically, how is the mRNA built in the nucleus? So your mRNA actually has a protective cap, a poly A tail. That means the tail is AAAAA, or the base adenine. Now you do have non-coding regions or introns, and you have coding regions called exons. What happens is that before the mRNA is fully developed, these introns or non-coding regions are spliced out. So you remain with the cap, exons, and the poly A tail. Eventually, that will complete transcription. Translation is where we actually get the formation, the transfer RNA brings the specific amino acid for the specific codon, and that's how we get translation to eventually have the specific amino acid.